Welcome back to Bio 43 Pathophysiology, Lecture 2, Mechanisms of Cell Injury. On the previous video, I talked about adaptive changes that cells go through, either normally or as part of a pathology. Now I'm going to discuss some specific mechanisms that cause cell injury, which in turn may cause adaptive changes as a result of the injury. But before I do that, I should mention that cells may undergo reversible injury, as in able to recover, or irreversible injury, meaning that they die. Examples of cellular injury that may be reversed if the injurious stimulus ceases and the injury is not too extensive include cellular swelling, loss of ATP, and detachment of ribosomes. Cellular injury that results in severe vacuolization or totally emptying out of the mitochondria, however, would be an irreversible example. The term necrosis is often used to describe pathologic cell death, while apoptosis is used to describe normal, genetically programmed, and timed cell death. We'll discuss these terms later on in the presentation. Overall, there are four different mechanisms of cell injury and cell death that occur regardless of the type of cellular insult. 1. Defects in membrane permeability allow for the loss of essential enzymes and the influx of calcium, which damages the mitochondria. Secondly, ATP depletion leads to inactivation of the sodium-potassium pump and subsequent decreases in protein synthesis, causing cellular swelling and loss of membrane integrity. Third, increases in intracellular calcium leads to elevated intracellular enzyme activity. And the fourth of the common mechanisms of cell injury is when free radicals cause damage to the cell membrane, alter intracellular protein activity, damage DNA, and alter mitochondrial function. Each of these causes listed here involve one or more of the four common mechanisms of cell injury, so we'll start by discussing hypoxia. Hypoxic injury is the most common type and damages the cell through the ATP depletion mechanism. Oxygen deprivation slows and eventually halts, and the production of ATP in the cell's mitochondria, which causes the cell's ATP-dependent pumps to fail, like sodium, potassium, and calcium. Okay, this creates an accumulation of ions. In the case of too much sodium in the cell, excess water enters the cell by osmosis, causing the cell to swell. A buildup of intracellular calcium in the mitochondria further inhibits them, and a small amount of ATP are produced by anaerobic glycolysis, which produces lactic acid as a byproduct, lowering the cell pH in the process and further inhibiting enzyme and protein function. Notice the last statement on this slide. Internal enzymes can be measured in lab tests to estimate cellular damage. Now we're getting somewhere. You already knew that, but now you know the reason why we can order enzyme tests to diagnose different clinical diseases. Can you think of a condition where we order enzymes in the blood that tells us if someone has the condition? No worries. If you can't at this point, we'll talk about it a lot next week.